Hello students, welcome to the biology class. Today we are going to continue with our chapter Respiration in Plants. In our previous session we discussed about the common pathway of aerobic and anaerobic respiration, that is glycolysis, and today we are going to discuss about the steps involved in aerobic respiration. The events in respiration: in aerobically respiring cells, pyruvate acid produced at the end of glycolysis from cytoplasm enter the mitochondria. Now the site of aerobic respiration is mitochondria. It initiates in cytoplasm and complete in the mitochondria. In aerobic respiration, complete oxidation of pyruvate acid by stepwise removal of all the hydrogen atoms with the release of 3 molecules of carbon dioxide takes place. The transfer of electrons removed as a part of hydrogen atoms to the molecular oxygen along with the ATP synthesis takes place. The main key or the events of the respiration complete in 2 steps, and they are Krebs cycle or TCA cycle, electron transport chain or electron transport system inside the mitochondria. So the 2 main events which complete the aerobic oxidation of pyruvate acid, they are Krebs cycle and electron transport chain or electron transport system. Gateway step to the Krebs cycle. This is important step, that how the pyruvate acid enter into the mitochondria. Pyruvate acid is oxidatively decarboxylated into 2-carbon acetate unit which joins coenzyme A to form acetyl CoA. The reaction is catalyzed by the enzyme pyruvate dehydrogenase and requires magnesium ion, thymine pyrophosphate and lipoic acid. NAD is also reduced to NADH plus hydrogen ions. Acetyl CoA is the connecting link or connecting reaction or gateway step between the glycolysis and citric acid or TCA cycle. This is the sum up equation of gateway step. This is pyruvate which is synthesized in the cytosol, that is cytoplasm, during the end of glycolysis. Then it enters into the mitochondria and the reaction is catalyzed by the enzyme pyruvate dehydrogenase, and NADH is reduced to NADH plus hydrogen ions, and this is release of CO2. So this is decarboxylation takes place and coenzyme is being added to form the acetyl CoA. This is again your 2-carbon compound. The next and the very important step of aerobic respiration is Krebs cycle or tricarboxylic acid cycle. This cycle has been named after the German biochemist in England. Name of that biochemist is Sir Hans Krebs. So the name is Krebs cycle. Krebs cycle is called as citric acid cycle because the first stable product of this cycle is citric acid. It is known as TCA or tricarboxylic acid. Since the first product, that is citric acid, has 3 carboxylic group, hence it names TCA or tricarboxylic acid. The site of Krebs cycle, it occurs inside the matrix of mitochondria. Each step is catalyzed by specific enzyme. it results in stepwise removal of hydrogen, and that hydrogen is received by coenzymes NAD and FAD to form NADH2 and FADH2 respectively. The stepwise formation of Krebs cycle. So the first step, that is the gateway step, acetyl CoA, that is acetyl coenzyme A, that is your linking reaction. It enters the matrix of mitochondria with the help of 4-carbon compound, that is oxaloacetate, and they produce the 6-carbon compound, that is citric acid or citrate. So this is the first stable 6-carbon compound, hence the name is citric acid cycle. In the presence of enzyme, that is citrate synthetase, and CoA is set free. The second step is the citric acid that change into the cis-A kinase with the removal of the water, and the molecule of water is added to cis-A kinase to form the isomer of citrate, that is isocitrate. Fine. So this isocitrate produced in this reaction is dehydrogenated. A pair of hydrogen atoms are removed, and that pair of hydrogen is accepted by NAD to form NADH2, and this reaction is catalyzed by isocitrate dehydrogenase, and this also shows the removal of carbon dioxide. The next reaction, that is your alpha ketogluteric acid. So this is alpha ketoglutarate is formed. The alpha ketoglutarate undergoes the decarboxylation as well as it will undergoes the dehydrogenation. So the two steps takes place in this reaction, that is addition of CoA. Along with that, the removal of hydrogen ions accepted by the NAD to form NADH2, and the CO2 is released, and they form the another substance, that is succinyl coenzyme A, in the presence of enzyme alpha ketoglutarate dehydrogenase. Now this alpha ketoglutarate dehydrogenase, which convert the alpha ketoglutarate into succinyl CoA, undergoes the dephosphorylation, and they form the succinate. And during the dephosphorylation, the phosphate is being taken by GDP to form the GTP. So that is GDP is guanosine diphosphate and GTP is guanosine triphosphate. So the formation of direct ATP takes place in this step only, in Krebs cycle or in citric acid. Now this succinate undergoes the dehydrogenation in the presence of enzyme succinate dehydrogenase, and the hydrogens, they are accepted by another coenzyme, that is FAD, to form FADH2, and they result in the formation of fumarate or fumaric acid. So next is the formation of malate from the fumarate in the presence of enzyme fumarase by the addition of water is there, and they form the malate, or the another name of malate is malic acid. Now this malic acid or malate undergoes the dehydrogenation. Again that hydrogens are accepted by NAD to form NADH in the presence of enzyme malate dehydrogenase to form the oxaloacetate or oxaloacetic acid, and then the cycle goes again. So in this cycle, so first acetyl CoA in the presence of enzyme citrate synthetase with the removal of coenzyme A combines with oxaloacetic acid or oxalocitrate acetate to form the first stable compound, that is citric acid or citrate. Then they undergoes the isomerization to form the isocitrate. Isocitrate undergoes the dehydrogenation in the presence of enzyme isocitrate dehydrogenate to form the alpha ketoglutarate. This alpha ketoglutarate they undergoes dehydrogenation in the presence of enzyme alpha ketoglutarate dehydrogenase as well as the decarboxylation by removing the CO2 and addition of coenzyme A, and they result in the formation of succinyl coenzyme A. The succinyl coenzyme A undergoes the phosphorylation to form the GTP in the presence of enzyme succinyl coenzyme A synthetase. CoA is released and we form succinate. Now this succinate again undergoes the dehydrogenation. That hydrogens are received by another coenzyme, that is FAD, to form FADH2 in the presence of enzyme succinic dehydrogenase, and they form fumarate or fumaric acid. The fumarate or the fumaric acid will form the malic acid with the addition of water, and the enzyme is fumarase. Now malic acid again undergoes the dehydrogenation in the presence of enzyme malate dehydrogenase, and NAD is converted into NADH. Now oxaloacetic acid form. Now again the reaction will continue and the cycle will continue. So in this cycle you can see NAD 1, 2, and at the 3 steps the reduction or dehydrogenation takes place, which is received by NAD to form the NADH2, and at one reaction FAD is reduced to form the FADH2, and there is only one reaction where the direct synthesis of ATP takes place. So this is very important cycle in which we get a maximum of the ATPs. Significance of Krebs cycle. This is the major pathway for the formation of ATP molecules, and many intermediate compounds of this cycle are used in the synthesis of other biomolecules like amino acid, fatty acids, and their building blocks or the macromolecules like proteins. So this is very important cycle, very important according to the examination point of view. Please go through this, copy it out, practice the cycle again and again for your exams. In our next session, we will discuss the electron transport chain, in which we discuss that how the NADH2 hydrogens are being converted to give you the energy. Till then, stay home, stay happy. Thank you and have a nice day.